In this video, we're going to discuss what a cell culture is, its application, types, the difference between proliferation and differentiation, as well as scaffolds, including their engineering and decellularization. Let's start by defining what a cell culture is. So, a cell culture is basically removing cells from an animal or a plant and growing them in a favorable artificial environment. So the cells can be removed from the tissue directly or desegregated by enzymatic or mechanical means before they're cultivated, or they can just be gotten from a cell line or strain that's already established. We're going to talk about the applications of cell cultures. There are many useful applications of cell cultures, including studying the physiology and biochemistry of cells, such as the metabolic studies or even the process of aging. We also can study the effects of drugs and toxic compounds. We can study mutagenesis and carcinogenesis. We can do drug screening and development. And finally, we can manufacture or produce biological compounds, such as vaccines or therapeutic proteins. There are types of cell cultures. Let me start by the basic ones. We have the primary cell culture. In the primary culture, it's the culture stage after the cells are isolated from the tissue and proliferated under the appropriate conditions until they occupy all of the available substrate. This is called confluence. After the cells reach the stage of confluence, they have to be subcultured by transferring them to a new vessel with fresh growth medium to provide more room for continued growth. This is where secondary cell culture comes in. It's basically a subculture of the primary culture. So when cells in the primary culture occupy all of the available substrates, or if they're in suspension and they exceed the capacity of the medium to support more growth, cell proliferation starts to reduce or stop. To continue the growth and proliferation, the primary culture has to be subcultured. This process is called secondary cell culture. Now let's talk about the difference between proliferation and differentiation. So proliferation is basically done to increase the cell numbers without changing any behavior. Undifferentiated stem cells have the best chance to expand in the laboratory, but transplantation may need the cells to be differentiated or have the potential to at least differentiate. Whereas in differentiation, it is the change in cell behavior and characteristics without a major change in cell numbers. The differentiated cells in a tissue have a limited ability to proliferate, so differentiated cells don't form primary cultures unless special conditions are used to promote their attachment and preserve their differentiation. Let's talk about scaffolds. Scaffolds are basically a material which substitutes the extracellular matrix just temporarily. So what is the extracellular matrix? It basically helps in homing and the aggregation growth, and the differentiation of the cultured cells. So the idea of tissue engineering is to repair damaged or diseased tissue, which is done by regenerating the tissue in some way. Most of the cells are attached to an extracellular matrix, and the composition of that matrix really depends on the type of tissue that's involved. So, for example, with bone, it can be collagen and a calcium mineral, whereas with skin, it can be elastin and another mineral. So in order to put something in the body and have the cells attached to it, it has to be made in a way where cells can migrate through it, attach to it, differentiate and proliferate, just as they would in a regular body. As they perform their normal functions, they start to secrete the natural extracellular matrix and the engineered scaffold is reabsorbed. So, there must be a balance between the rate at which scaffold resorbs and the rate at which the cells are depositing the new native extracellular matrix. So, scaffold resorption, there has to be a balance between the rate of cells depositing the new extracellular matrix. Now we're going to talk about scaffold engineering. There are 5 important factors to consider during scaffold engineering. They are biocompatibility, biodegradability, mechanical properties, scaffold architecture, and manufacturing technology. Let's start with biocompatibility. So the cells have to adhere, function normally, and migrate onto the surface and eventually through the scaffold and start to proliferate before they lay down a new matrix. After implementation, the scaffold or the tissue construct has to ignite some sort of small immune reaction so it can prevent it from causing a severe one, which can reduce the healing or cause some sort of body rejection. The second one is biodegradability. So the whole point of tissue engineering is to allow the body's own cells to replace the implanted scaffold or tissue construct, so they have to be biodegradable so that the cells can produce their own extracellular matrix. The byproducts cannot be toxic, and an inflammatory response and controlled infusion of cells like macrophages is sometimes needed so that the degradation can occur with tissue formation. Thirdly, we have mechanical properties. The scaffold should have mechanical properties that are consistent with the anatomical site into which it's being implanted, and it has to be strong enough to allow surgical handling during the implantation. The fourth is scaffold architecture. So scaffolds should have an interconnected pore structure and high porosity for cellular penetration and nutrients diffusion to cells within the construct and to the extracellular matrix formed by these cells. Porous interconnected structure is needed so water can diffuse out of the scaffold and products of scaffold degradation have to be able to exit the body without interfering with other organs and surrounding tissues. The last one is manufacturing technology. For a scaffold or tissue engineered construct to become clinically and commercially available, it has to be cost effective and should be possible to scale up from making just one at a time in a research lab to a small batch production. Now regarding scaffold decellularization, depending on the tissue being processed, a decellularization strategy can include physical, chemical, and enzymatic treatments, all just to remove cellular antigens while also preserving the composition and organization of the native extracellular matrix. I hope you were able to learn about cell cultures and scaffolds. please consider subscribing to this channel for more videos of this nature. Thank you for watching.